on blurring the lines of research and public health with SARS and CoV-2, genomic sequence surveillance.
All right, thank you, Brian. And I'd like to thank everyone for joining us. And I'd especially like to thank Jason, who's helping with the Zoom and the AV tech today. What we are going to be doing during this panel discussion is talk about genomic surveillance, which is a form of enhanced testing where, in addition to knowing whether or not someone is infected with SARS-CoV-2, the virus that causes COVID, we determine the sequence of that virus. And that allows us to understand more about the virus than we can learn from testing alone. However, the fact that we're learning more about the virus than just what we learned from testing alone also introduces some new complexities and some new challenges. And the point of today's panel is to really talk through some of these challenges and some of the dimensions in which they exist today and how we might try to improve them in the future. And so I'm really delighted to have a panel joining me today that has a broad breadth of experience across academic research as well as local and state public health. And so what I'd like to do is have each of the panelists introduce themselves and their research very briefly. And then what we're going to do is we're going to have an open-ended panel discussion that is hopefully going to be fully interactive. So I would really build on what Brian said and encourage you to ask questions in the chat, provide feedback to what you're hearing in the chat. And if you're so inclined, we'd be happy to even have you use the raise hand feature in Zoom, unmute, turn on your video so that you can contribute to the conversation as you see fit. So with that, let's start the introductions by first asking Dr. Kelsey Florek to introduce herself.
Thanks, Dave. So I'm Kelsey Florek. I'm the Senior Genomics and Data Scientist here at the Wisconsin State Laboratory. And my role here at the laboratory is really looking at infectious disease genomics across the board. So before the pandemic, we were heavily invested in antimicrobial resistance, looking a lot at different bacteria causing issues in clinics and facilities across the state, monitoring really a lot of different mechanisms and modes of resistance travel and things like that. And so we were really interested in trying to capture a good understanding or a better understanding of what's out there and what's happening in Wisconsin and comparing that to the national picture. Once COVID came along, we kind of shifted gears and really started pushing a lot of our efforts into better understanding the pandemic as it kind of continued on and understanding how and what variants were emerging within Wisconsin and where they were coming from. And so we've kind of worked together really closely with Dave and Tom and the AIDS Vaccine Research Laboratory, as well as several other laboratories across the state, trying to really emphasize and push forward these new ideas of how we can collaboratively add to the genomic surveillance that's occurring.
Great, thank you, Kelsey. Brittany Grogan is joining us, even though she is masquerading, I think, as Cat Grande. Brittany, would you like to introduce yourself?
Yeah, sorry, I am Brittany Grogan, not Katerina Grande, who is my colleague. I don't have the ability to change my name on here. I don't know if anyone as a host does, but anyways, I am a data analyst with Public Health Madison in Dane County, currently serving as the lead of our COVID data team. Formerly, Kat, my colleague, was our lead, and she is out on parental leave right now. I've been on our data team since the beginning of the pandemic, so 2 years now. And I've been involved in the sequencing stuff as I've kind of been the main COVID data person responsible for tracking outbreaks and clusters of cases in Dane County. And the exciting thing with sequencing has been the ability to identify transmission clusters. So like being able to tell whether people's infections are linked, like if one of them infected the other, which has been able to tell us a lot about where and how transmission is happening. There are lots of exciting possibilities with even more robust sequencing, and it's been exciting to be a part of this.
Great, thank you, and thank you for joining us. Tom, would you like to go next?
Sure, I'm Tom Friedrich. I'm a professor in the School of Veterinary Medicine, and my lab is interested in the ways that viruses start to infect new hosts and become transmissible and cause disease in new hosts. And so we use a number of approaches to address these questions, but a lot of them we ask through an evolutionary lens. And so we use viral genomics to try to understand the evolutionary processes that viruses have to go through to do these things. So my lab has been interested in influenza since we started back in 2008, and then we started studying Zika virus when that emerged back in 2015, 2016. And then I think that placed us well to start a collaboration to look at the spread of SARS-CoV-2 through space and time here in Wisconsin in collaboration with Dave and Shelby. And as Dave knows well, we have been working together for, like almost 25 years since we were PhD students together in the same lab. And so we've done a number of collaborative projects, in all of that time, including all of the work that we'll discuss today on SARS-CoV-2.
And lastly, Dr. O'Connor, would you like to introduce yourself?
Hi, I'm Shelby O'Connor. I'm a professor in the Department of Pathology and Laboratory Medicine. And my lab, we study host immune responses to different infectious pathogens, primarily HIV and tuberculosis. But for the last couple of years, I sort of pivoted some of my work to focus on working with Dave and Tom to perform surveillance for COVID, which began with deploying a mobile saliva testing program, and then teaching over 100 schools how to do antigen testing for SARS-CoV-2. And then in the last 6 to 8 months, deploying air surveillance instruments to different settings to look for a virus that might be circulating in the air.
All right, thanks Shelby. So let's start with some basic stage setting questions. And so I'm sure everyone here has heard of Omicron, and before that, Delta and Alpha and Beta. And so these are different variants of concern that the World Health Organization has designated. And we identify whether a virus is an Omicron virus or a Delta virus through this process of genomic sequencing. Just so that we're all on the same page, Kelsey or Tom, would you like to just briefly describe what goes into a sequencing experiment? So the equivalent of the after-school special about how a bill becomes a law. How does a Q-tip in your nose become a sequence?
Do you want to do that, Kelsey, or should I?
I can start us off, and then you can come back and fill in gaps from your perspective.
Perfect. Okay.
So it really starts with a test. So in the clinic, you go in, you have your swab taken, and PCR performed to determine if that sample is positive or negative. Once a positive sample is determined, we then take that sample and that virus in that sample. Basically, we break the genomic information out of that virus and fragment it into a bunch of chunks. And the process of fragmenting can be a little bit different depending on how or what approach you're using. But basically, we're trying to take all of that genetic information, break it into a lot of smaller chunks, and then get the genomic sequences of all of those smaller chunks kind of in a high throughput approach where we sequence everything all at once. And we do this across multiple samples as well. So each sample is tagged with barcodes that we're able to differentiate them. And then once we have that sequencing data from that run, we essentially have all of those fragments en masse sequenced in a data set. So you can kind of think of this as if you took a bunch of novels off your bookshelf, put them through the paper shredder, and then tried to reassemble that data in the computer. And it's very computationally intensive, but luckily there's a lot of nice algorithms that have been pushed forward in this area of bioinformatics to kind of make that process a little bit quicker. So once we basically put that genome back together and we have that full picture of what that virus looked like, then we start comparing that virus to other viruses that we've previously seen to better understand how that virus fits into the overall pandemic structure. And there's a couple of methods of doing that. So one is to basically take that sequence and compare it to a set of pre-chosen reference sequences. And from there, we can gauge if it's a new virus or if it's something similar to Omicron or something like that. And then another method that's been kind of really used in this pandemic has been a machine learning approach, where basically it's a method of classification where we try to rapidly compare that structure to a pattern of structures that's been predetermined or pre-learned. And then from there, we can use a taxonomic identification that way. So when you hear of things like BA1 or BA2, that's actually where that comes from. It's that machine learning algorithm form of classification.
So you mentioned BA1 and BA2. So for those of you who don't follow the nomenclature, those are two different sublineages of Omicron, one of which, BA.2, has newly arrived in Wisconsin just over the last couple of weeks. And so it provides a good example of thinking about what's the value and what are the limitations of this kind of sequencing approach. So I think it would be helpful to get both an academic approach, and maybe Tom, you can handle that, but also a public health approach. So Brittany, like, how worried should we be about this BA.2 variant? Like, what do we do with this information if there is a new variant that we detect by sequencing?
So how about we talk a little bit about how we detect that from an academic perspective and like quantify how much of it is around. And then Brittany can tell us like whether we should be worried about that in the real world. So picking up from where Kelsey left off, you have these a bunch of samples of people who've tested positive for COVID. And then through the process that she characterized, you characterize the genome sequences of these viruses, and you can say, based on like a list of mutations that are only present in Omicron, like, is this an Omicron virus or not? And put those in these categories. These are Omicrons, these are Deltas, these are on the Omicron rubric, these are BA1s and these are BA2s. But you can also take that information a step further, a couple of steps further. And first of all, you can begin to draw family trees that link together all of the sequences that we have in Wisconsin with all of the sequences that are being generated worldwide. So just last week, we heard that there were almost 8 million sequences of distinct SARS-CoV-2 samples that have been characterized by different labs throughout the world. All of this information is pooled more or less in real time in a global database and then made into a global family tree. So we can begin to say, okay, the viruses that are circulating here in Wisconsin, these BA2 viruses, they look like viruses that came from, I'm going to make this up, this isn't really real, but like, let's say Denmark. So, they look very closely genetically related to viruses that came from Denmark. And so that suggests that perhaps someone traveled to Denmark and then traveled to Wisconsin, and that is how this particular virus was introduced into Wisconsin. We can also ask, if we see BA2 cases, numbers of cases growing locally, and we're seeing more infections attributable to this BA.2 virus, the phylogenetic tree, this family tree, can tell us, is that one big outbreak that maybe started with one introduction from one place? Or are we seeing sort of multiple concurrent smaller outbreaks that might have had different origins, different starts, maybe introduced from very different places in the world? And that can have some implications for how you think about responding to ongoing outbreaks and then stopping future outbreaks from happening. At least that's what we like to think in academia, but I think maybe Brittany can tell you in the real world how you use this information.
So Brittany, if Tom's example of something rotten in the state of Denmark is true, what do you do with that when it arrives here in Madison?
Yeah, so we did find out recently about our first BA.2 case, which was in Dane County, the first known one in Wisconsin as well. We now have at least 3 in Dane County, maybe more, I think at least 7 statewide. So it's always a little hard right at the beginning to know kind of what is happening when there's that few of sequences identified. But I know for us in Dane County, things that we've been thinking about is looking at us compared to other places in Wisconsin. So kind of throughout the pandemic, Dane County has had comparably a pretty low case rate to other places in Wisconsin, which is impressive considering we've also done, I think, like the most testing, or at least comparable to Milwaukee, which has like 3 times the size of our population. So we pretty much generally had low case rates, low percent positivity. However, like during this Omicron surge, and now that Wisconsin is kind of coming down from this Omicron surge, we've kind of been on the higher end of case rates. It seems like our decrease in cases has not been as rapid as some other places in Wisconsin. So something we've been thinking about is maybe, is there more of this BA.2 variant spreading? Is that maybe here in Dane County, like did it hit us first before it's gotten to other places in Wisconsin? Is that slowing our descent a little bit? It also is just, there's always like lots of things happening at once. You can never pinpoint one thing. So there's the introduction of the BA.2 Omicron subvariant. There's also a few weeks ago, UW came back. So we had a bunch of students come from all over the place back to Madison. You can, on our data dashboard, which we update on weekdays, you can see that in pretty much all age groups, there's a really clear descent from the Omicron peak. But in that 18 to 22 age group, there's a tiny little like increase recently, which you don't see in the other age groups. So there's that. And so it's kind of all these things together where there's just a lot of things happening that might be slowing our descent in cases. And so we try and take in all of that information and kind of figure out what we should do, what's going to happen in the future. So that's something we're still discussing now, but it is certainly possible that the BA.2 is part of this.
So I have a little bit of inside knowledge here because Tom and I generated those BA.2 sequences, and I know that that this is a question that I would like to put to maybe Shelby and Kelsey. You know, there's like a 10-day lag between when we got those samples in the lab, when the samples came out of someone's nose, until we had the data in these global repositories and went through all those steps that Kelsey outlined. And so there's, you know, a lag in time between when something arrives and when we're aware of its sequences. What is the timing that makes sense? Like, how fast does it need to be for it to be usable? I think if we said, well, we find out a year later, that's not going to be helpful, which means that there is a stopwatch time beyond which this kind of data is not helpful. But how quickly does it need to be generated in order for it to be useful? Shelby, would you like to take a stab at that first?
Sure. I think as much as the public would like to know, if they're walking around, is there a virus floating past them that might, be that BA.2 that infects them? And then, that's the one that's in their nose. I think that being able to obtain that sort of real-time information is just not feasible technically to ever happen. And furthermore, I think that knowing that information can lead to reflexive reactions sometimes that are too fast and maybe should be thought about more deeply before actually responding to them. So, I mean, I think that the sort of, it does depend on the infection, right? In this case, we're talking about a pathogen that infects people and, usually gets cleared in a couple of days. And so for this particular disease, I feel like we're at a pretty good pace where we see people getting infected. We kind of learn within a week, 10 days, 2 weeks, what that pathogen looks like. We can then be able to reflect on what's going on in the population at large. But if like this pathogen was a much more delayed infection, then we would, we'd be okay if it took a little bit longer. But if it was an even shorter infection, we may need to speed it up more. I think we've hit on a reasonable place for this particular pathogen to learn about what's going on around it.
I'm not sure if Shelby's frozen for anyone else, but she's frozen for me. I'm seeing some thumbs up. So would anyone else like to tackle that question too?
Well, this kind of, I can build on a little bit on top of what Shelby was kind of putting out there. I think there's a couple of things to consider too, is what's important for a person versus what's important for public health, where, like what matters is when we think about the Omicron response between BA.1 and BA.2 clinically, your responses should be the same. Like there's not going to be anything drastically different in how a person is cared for that has BA1 versus BA2. There are particular instances where that may change in the future, but that's something that needs to be addressed with different therapies and different vaccines coming out. But with public health, I would say that the time frame that we're able to sequence things is actually really valuable, because we're able to see in near real time how these things are changing over time. We're able to better understand like these travel associated cases. When we start to see a particular outbreak or something like that, we're able to start looking at the sequence data coming from those cases and start matching and start, informing different recommendations, whether we need to do mask mandates or lockdowns or anything like that. Like case counts help guide that, but so does knowing what variants are out there. That helps guide those practices as well. And then another layer on top of all of this is, we're essentially creating the fossil record for this pandemic with every sequence we generate. And when you think back to like the 1918, like we had to dig samples out of the permafrost to be able to get any sequence data out of that. Whereas here we have probably the most sequenced virus in existence, just because of the massive effort that's gone into this pandemic. And so we are going to be learning things about this pandemic for the next several decades.
Speaking of permafrost, I think Shelby is now unfrozen and had another comment.
Sorry, I have no idea why I froze up, but I did have one more comment about another reason. The question about timing, there's one case where it is really important for us to be able to know what a person's infected with if we could act upon it. And, some people probably are aware that for those infected with Omicron, we're able to be treated with, one monoclonal antibody, but not other monoclonal antibodies. And so knowing whether or not people who came in the hospital were infected with an Omicron variant versus a Delta variant, that becomes an important and a clinically meaningful piece of data for someone to have on hand. And so in those cases, it would be really great to get data back as quickly as possible so that those clinical decisions could be made. The challenge with that, though, is that trying to generate that data in a way that is allowable, I guess we'll just say, through CLEA mechanisms is not doable in the current climate. So, trying to come up with ways in which we can identify sequences in a way that are clinically meaningful, I think would be a major step forward.
Well, let's try not to use acronyms that everyone might not be familiar with. So CLEA is the certification group that manages accreditation of diagnostic labs. And I think it brings up a point that a lot of people find confusing that we should spend a little time on, which is that the rules that are in place for what can be done diagnostically were authored in a time when genomics was either non-existent or quite primitive compared to where it is today. And so if we generate a sequence in our lab, or if Kelsey generates a sequence in her lab, we can't turn around to a doctor and say, right, so patient Lindsay has an Omicron virus that's not going to respond to these antibodies. We're not allowed to do that because of the rules that are in place. And that can come across to people as very frustrating. We have the ability to generate this data. The data can be used to inform decision-making, like what monoclonals work. And yet that data is not allowed to be used to do that, given the constraints that Shelby talked about. So what do others on the call think about this? And what do people on the panel think about this? Is this okay? Is this the way it should be? Is this something that we should try to move beyond? Tom, you look like you're aching to say something.
I wouldn't characterize it as an ache, but I will say it anyway. Well, I think that there's sort of two things here, right? So there's an understandable desire on the part of regulatory agencies to make sure that if some sort of laboratory process, like some sort of laboratory experiment, is going to be used to make decisions about patient care, or data from that is going to go into a patient record, that we have, like that is as credible information as possible. Like if you, if you test positive for a pathogen and you begin to be treated and you don't really have that pathogen, that could be a really big problem. So like there's an understandable desire to regulate these processes. On the other hand, I think, as you say, there's a gulf between the world in which the regulations were initially made and the world in which we live today, where you can have fast, reliable genomics. Whereas, as Shelby said, as Omicron is emerging and patients come to the clinic with severe COVID, you have a pharmacopoeia of monoclonal antibodies that could treat them if they had Delta. But most of the monoclonal antibodies that you have in your medicine cabinet in the hospital, you already know from prior experience in other places, they're not going to be effective against Omicron. So I could give them to my patient if they had Delta, but I shouldn't give them to my patient if they have Omicron. And then here's a lab that can give me this information, but they're not allowed to give me that information. And yet we can feed the same information to public health and they can say there's this many Omicron cases, in Dane County, and track the spread of Omicron in time that way. So the data is useful, but it's not being used to its full utility. And I think that, you and I have both had this experience in Shelby too, and I'm sure Kelsey also, throughout the pandemic of pushing against sort of older regulatory regimes that were not built for responding rapidly to a pandemic with all the tools that we have available today.
So just to push on that a little bit, so what changes would you make if you were ruled the emperor of rule changing for a week?
Well, I think that it is tricky because we also, this is going to be kind of inside baseball about sequencing, but sequence data can be tricky to get right. And so, we've talked a lot about data standards with the national group from the CDC. And so there are technical barriers sometimes to getting appropriate data. And if you don't know what to do with that, so for example, there have been cases where, groups cannot get the full genome sequence of a virus. So, they have a sample, they've successfully characterized 80% of the nucleotides of the letters in the viral genome in that sample. And they can't tell from their data what the other 20% are. What do you do with that? There are some groups who decide, well, in order to get this into a database so that I can share my data, I'm just going to fill in that missing information, with kind of a known sequence to which I would compare the reference sequence that Kelsey was talking about earlier. That is obviously not okay if you are trying to characterize the virus that a patient is infected with in order to drive treatment decisions. So I do think that there have to be some kind of minimal standards, but I think that there's a clear pathway to get there that would not necessarily require all of the onerous nature that you would sort of have from existing regulations. I'm not going to pretend to be an expert in all of the regulations of CLEA, as Shelby was talking about. So I can't stand up here and make a specific prescription. Maybe Kelsey is in a better place to do that.
Yeah, I can highlight a couple of points. I would say, for a laboratory looking to do what's called an LDT, or laboratory developed test. So this is a test that could be used to report it back to a physician or clinic or primary care or something like that. It has to go through a pretty onerous process. And something as simple and straightforward as a PCR test that is in its nature not as complex as sequencing is still quite a lift for a laboratory to validate in under the guise of regulation. And I think there is a lot of value in how labs approach that and making sure that things are done in a reliable and reproducible manner, and that there's lots of tracking and logging and making sure that the results that are coming out of the laboratory are accurate enough that you can make informed clinical decisions. And I would say probably one of the biggest things that's lacking right now is really clear and good guidance on how to approach validation of any sequencing-based test. There's been a number of laboratories that have pushed that forward in areas not related to coronavirus, but it's often an extremely onerous process that involves a lot of what I would kind of say is archaic approaches to understanding sequence data, where there's a lot of use of positive controls on every run to make sure sequencing runs appropriately, which, when you've done enough sequencing, that there's a lot of information that comes out of that tells you how good or bad that sequence data is. And so there just needs to be, sequencing is still a relatively new tool in public health. And so it just needs, I think, some time before there's a clear understanding and guidance on how to push that forward.
So Kelsey, I'm going to stick with you for a second because we have a question from Denny in the chat that asks, are all positive samples sequenced? And so Kelsey maintains a statewide dashboard that looks at this very question. So would you like to comment on that?
Yeah, so I just pasted the link to the dashboard in the chat, so you can go and check it out. This is kind of a running total of all the sequences that have been generated for the samples collected in Wisconsin. So roughly, I would say, I think we're around 4 to 5% of all cases being sequenced. That might have dropped a little bit with the recent Omicron peak. Our effort to sequence, it's still a very expensive process, and it would be incredibly expensive for us to sequence every positive sample. But especially when case counts tend to spike like they have recently. But what we have tried to do is do a more geographic and representative approach, where we're capturing information from across the state and across as many counties as possible. So that way we have a good picture of what's happening both in metropolitan and in rural communities. And again, that kind of just helps feed more information to the Department of Health and local public health departments as they start to think about guidance and practices and interventions going forward.
So Brittany, I'm going to turn it over to you. So the question is, are all sample sequenced? But the word all really kind of needs to have an asterisk next to it, because when we choose a denominator for samples that are sequenced, we're really usually using the number of PCR tests that are performed at laboratories like Exact Sciences to come up with that denominator. But in the last couple of months, people have become a lot more comfortable with the notion of at-home antigen testing, where those results might not even be reported to public health. And certainly those samples, if they're not reported to public health, are essentially invisible to the sequencing process. So can you comment a little bit on how antigen testing has changed the way that you have to think about data when you're processing it at the county level? Is there like a fudge factor for the estimated number of antigen tests being performed, or how do you handle that?
Yeah, so we've kind of had this issue throughout the pandemic. As we know, the suspected number of true cases is much higher than what basically anywhere has as like confirmed PCR positive cases. So in the beginning of the pandemic, testing was extremely limited, and we were using, so there's a case definition for COVID cases where a confirmed case is any case who tests positive with a PCR test. But we've also had this definition of a probable case. And a probable case is either one that tests positive with an antigen test that is that has CLEA oversight. So that'd be like one administered at a testing site. So a home test is not covered under this definition alone. The probable case definition also includes someone who has an epi link to another confirmed case. So they had close contact with someone else who tested positive, and also that individual is experiencing symptoms. So if they have symptoms and a link to a confirmed case, they can be considered a probable case. So we've tracked probable cases throughout the pandemic. And lately, the at-home tests have become a lot more ubiquitous. With those, we've kind of attempted to capture some of that data we have a survey that we can have people fill out to report their at-home tests to us. And so far, that's kind of just been our own like internal monitoring to have a sense of what the burden is out there. We have gotten hundreds of those reports, which we know is still an undercount, since definitely not everyone is reporting their positive at-home test results to us. But that's kind of helped us kind of gauge how many people are using those. But yeah, and then when we talk about sequencing, we can only request those who have a positive PCR test. So in the grand scheme of things, that's not capturing everyone who has COVID. It's only people who are seeking out testing, which is a subset, although in Dane County, since testing has been, I'd say, highly available and accessible at most times throughout the pandemic, except maybe during the worst of our surges. We have seen a lot of people get tested and feel like and we've had one of the lowest percent positivities throughout the pandemic as well in Wisconsin. So we feel like we've captured a good amount of cases. But yeah, the sequencing will only be a subset of all those true cases in the community.
So sort of we have a hand from Tom.
Yeah, I think just one thing to note here too is that sequencing can complement this ascertainment of cases through testing, and it hasn't really been done as much recently, but especially at the beginning of the pandemic when testing was really limited. You can use kind of evolutionary models to ask how many infections must there really be to account for the diversification in sequences that we're observing. So, with knowing kind of the mutation rate of the virus and the average time between infections, you can kind of integrate over the amount of diversity that you see over time in the sequences that you are collecting, and estimate how many cases must there really be to account for the genetic diversity that you're observing. So like these measurements can really be used to complement each other.
So another way you can use sequencing is, not by looking at the individual people who test positive, whether it's by antigen test or PCR. Now, Shelby, there's been a lot of talk in the news about wastewater, and you mentioned earlier air sampling. Can you talk a little bit about how environmental surveillance might complement this too?
So one of the benefits of environmental surveillance is you don't need someone to sign a sheet of paper and say that, they will allow you to swab their nose and be tested. Instead, you just collect something from the environment and you look to see if there is pathogen genetic material present in that sample. And so wastewater has been a frequently used source for environmental surveillance, where they're collecting samples from different sewers and isolating nucleic acids and looking for SARS-CoV-2. And I don't know, I should have put it up here. There's on the DHS site, you can actually look at the different wastewater sheds throughout Wisconsin and their detection of SARS-CoV-2 virus over time for the last couple months. Thanks, Brittany, for putting that up there. And so, there's a lot of value in that, because if you look at that data, you can see how the trends of virus detectable in wastewater have sort of been shaped, have a pattern that's very similar to what we're seeing in cases. There's a lot more virus detectable now in the wastewater because of the Omicron surge. And in fact, in some parts of the US, viruses were detected in wastewater sort of just preceding the detection of cases in the community. And so using something in the environment where people don't necessarily have to walk into a clinic to go get tested is a nice way to look for whether or not there is virus in our community without swabbing people's noses. Wastewater, however, is a somewhat noisy sample. You can imagine that stuff that comes through the sewer is full of lots of stuff. And so the tools that we have to use to look for viruses in wastewater sometimes cannot distinguish between what we're looking for and all that background stuff that's in the wastewater. And so those patterns, they do tend to nicely mirror what we're seeing for patient cases. There's a lot of noise. And so as Dave mentioned, we've been trying to use instruments available from Thermo to look for virus in the air. And so we have these different samplers, which are like reverse hair dryers. So they're basically sucking in air, and they suck in air. And as they do that, they take pathogens and collect them on a cartridge inside the instrument. And we can look to see if there are pathogens that we have collected from the air in that instrument. And this is a nice option to look for sort of localized, we can locally capture virus from the air setting. So for example, you could take a sampler and put it in a school, or you can put it in a cafeteria, or you could put it in an airport near the baggage claim, or you could put it on a jet bridge or in a bus. And you could look to see if there's virus present in the air in those small locations. Now, of course, this gets back to that topic we were discussing before. How quickly can you find out if there was virus present in the air in that environment in which you are living? And that's a much bigger challenge to follow. So similar to wastewater surveillance, what we can do is just sort of look at the patterns for whether or not there are respiratory viruses in our environment without basically looking at individual people. I hope I kind of gave a reasonable summary over the power of sort of environmental surveillance, and that it's a great thing to do without having to swab noses, but gives you a nice picture of what's going on in our community.
Sure. So a question then to everyone is, okay, so these are all, we think, useful things, at least to a first approximation. But where do you stop? Like, it might be nice if we were sampling every sewer shed, every wastewater, if we had a network of air samplers in every room, a chicken in every pot, an air sampler in every corner of the country. But it's going to be expensive. We don't have the public health infrastructure to handle nearly the volume of data that would entail. You could argue that we've been underprepared even for the amount of sampling that we've done. We've really had to bootstrap that up. So when we're thinking forward a year, 3 years, 5 years from now, how do you find that balance of something that is large enough to be impactful, but sustainable enough so that it doesn't just crumble under its own weight?
I'll take a stab at that. I think this is actually a place where like interdisciplinary data science can really help us. So this is a place where modelers who can help kind of bridge between disciplines like epidemiology and environmental biology, environmental science, and viral evolution can sort of help us think about different scenarios that one could adopt. So, where is there a sweet spot between getting enough data in enough places of like these sort of nodes that Shelby was talking about, where we might sample a lot of people, that we can have kind of a reasonably good coverage of places where we might expect to detect pathogens that we'd worry about, and still have kind of tractable, more or less representative samples of what's going on. So like, do we need to sample every sewer shed? No. Can we identify sewer sheds that have attributes that we might like, that sample from a large number of people and that have populations that are, reasonably stable, so that it might make the signals a little bit less noisy? Is that going to work in more rural settings where they're like people don't have, like a big sewer infrastructure? That's probably like a city thing. What do you do to sample, less dense human populations where like a wildlife human interface might be more important than the spread of a pathogen from abroad into that community? Those are things that that modeling kind of exists to help us think through in a quantitative way. Like, what if we tried it this way? What if we tried it that way? And so I think that part of the the initiative for pandemic preparedness in the future is going to be just gaming out different scenarios, different surveillance scenarios, and trying to determine what is going to give us the most bang for our buck. I can't give you the answer to that question, but I think that's the beginning of the process.
So turning it to Brittany and Kelsey, from a public health perspective, one answer is make PHMDC 100 times bigger than it is today, and hire a whole bunch of PhD data scientists and modelers. Or you're going to have to find a sweet spot, or it's some combination there. But I think you were still going to find that there is going to be, skills mismatches. The skills that one has to do public health might not be the same skills that one needs to either do the sort of phylogenetics that Tom was talking about earlier, or doing some of the really advanced statistical modeling that he was just talking about. So how do you bridge that divide? Like, how do you grow public health in a way that makes it so that it can use this sort of data effectively, while realizing that there's not going to be infinite resources available?
So I can say from a public health laboratory standpoint, there's a lot of change happening. There's a lot of labs that are really realizing the value and benefit of sequencing and what genomics can do. At the start of this pandemic, sequencing was kind of not even really considered initially. It was more of just how can we get tests going and out the door. But then once we started to have variants emerge, a lot of laboratories started shifting into sequencing and trying to better understand. And now we're at a point where just about every state in the United States has a laboratory that's doing sequencing, a public health laboratory that's doing sequencing. And before that, a lot of sequencing was being done on foodborne because there's a very strong initiative through the CDC for foodborne illness and sequencing. But going forward, it's been kind of it's been hitting a learning curve, trying to bring this technology in and better understand it. And I think going forward, laboratories are going to continue to struggle and try to find ways to utilize this information. The amount of data that comes out of these sequencing machines is incredible. And it's not, laboratories aren't able to fully use the data to their advantage because of the missing skill sets that are in public health right now. Now, there's a lot of efforts to try to change that. There's APHL has several new fellowships trying to bring in skill and expertise in data analytics and bioinformatics. On top of that, there's a lot of laboratories that are pushing in different directions, and startup companies even that are trying to look at ways of leveraging the data that's being generated and benefit public health from that data. And I think going forward, we're going to see a huge change in how public health operates, kind of similar to how there was a huge change when PCR came along. And I think we're still wrestling with how that technology is going to be used and what's the most practical implementation. But across the country, I see more and more labs buying high throughput robotics, all kinds of stuff to support this effort. And I think the big part of this is going to be the workforce, and how do we get trained data scientists into public health.
You mentioned fellowships. How else do you think you should try to build and develop that workforce?
I would love to see some more programs that are partnered with the School of Public Health or things like that to kind of help guide some of these people that might be interested, that are early in their careers or early in their schooling. that might be interested in something like public health and kind of guide their thinking to answering those types of questions. But I think that would be really phenomenal.
So Brittany, as someone who is a data lead in a public health department, how did you get to where you are?
Well, I did both my bachelor's and master's in public health at UW-Madison. I was over at the state health department, DHS, for a few years before I came here to public health, Madison-Dane County. And I would love for us to have more data staff, more people in data science and laboratory science. I think there are a ton of opportunities. But what I see even like before we are able to hire those staff is really upgrading our public health infrastructure and data systems. I think those have been so neglected and underfunded, and we've just run into so many issues over the past 2 years with all of this lab data coming in that our systems were not built to handle. And we've had a lot of challenges with handling the amount of data and dealing with problems that arise with that amount of data. And we need more of a national investment in public health. It'd also be great to just have more interconnected systems, more federal systems, more ways that we can share data across county lines and across state lines, and have things just be a lot more comparable to each other. So I'd love to see that infrastructure be upgraded and improved, and then also see more staff to go along with it.
So let's unpack that a little bit more. So, I can pay my bank from my phone. I can, send communications around the world. I can talk over video with people anywhere. We have companies who have solved these really hard, big technical challenges at scale. Why do we have such a mismatch between what the existing systems in something as critical as public health look like, and the potential for what these systems could look like, given what public-facing systems look like? Why do we have this disconnect, and what do you think we could do to fix it?
I'm sure others have plenty of thoughts and ideas too. I think that in my experience, a lot of problems come from just, like I said, like not having nation level systems. And so states are left to either create their own systems or create their own things, or they contract it out to maybe a private company that doesn't have a lot of experience in public health and might not build it in a way that someone with that background and knowledge of public health would. And a lot of privacy issues where it's not always easy to share information between counties or between states, and dealing with all of the issues that arise from that can be a big challenge, I think.
Go ahead, Kelsey.
Yeah, I definitely want to add to that a bit. From a laboratory standpoint, it's been a huge challenge working with this level of sequencing data. Before the pandemic, we would do sequencing of outbreaks of 100 samples at most. And, right now we're at a point where we're sequencing 500 samples a week. And that level of change and that level of data has forced us to completely rethink how we manage and work with data. And so we've turned to cloud approaches. We're actually looking at planning out and developing a data lake approach to try to handle both the storage and analytics, but as well as the integration, because the sequence data is great to have, but unless you start to provide context for that data, it really is pretty useless. So once we start to add things like symptomology, hospitalization outcomes, like those kinds of things, then we can start to really start asking powerful questions of our sequencing data. And, you know, just earlier we were talking about skill sets, but I agree completely with Brittany. Like, our infrastructure has really been sorely lacking. And it's something I think we've all had to deal with across public health, where just the amount of data, the amount of samples, the amount of cases is something we've never had to deal with before. And trying to push that level of throughput through some of the systems that are existing has been a real challenge for everybody. And so I think going forward, like, yeah, I would love to see some sort of national approach. I know CDC is working on several different approaches for managing these things. But I think, you know, as new technologies emerge and things like that, and as costs come down for cloud computing, and as more opportunities come through, I think, you know, a lot of laboratories and states are going to be looking at different options for how to connect their data and how to make this stuff more meaningful.
If I can add an opinion as well, I was just thinking about this, and I guess I'm not a political expert on how money gets shuttled around, but at least as far as like the K-12 testing was concerned, when that was sort of emerging last year, it was my understanding that a small portion of those funds were actually going to the human capital involved in testing, whereas a lot of that was going to the testing itself. And so I think that when we guide our government to think about how to distribute funds, you know, it's easy to check boxes and say, I ran 1 million tests, or I, generated so many sequences, but it's much harder to say we trained this many people, or, you know, these many individuals were able to support these particular efforts, and that was a benefit, you know. And so being able to somehow, I don't know if it's quantify or be able to justify the use of funds to help support these activities that Kelsey and Brittany are talking about, and training people to support the infrastructure development, the data analysis, even just, providing the test to, say, kids in schools or whatever. Having that human capital is so important that should not be forgotten when thinking about the ways to distribute funds.
Well, if I could just also add something to tie together some of these threads, I think, there are things that we can do as scientists, and as Shelby was saying, there's kind of things we can do just generally as citizens. And so, what we've been hearing in comments from Brittany and Kelsey and Shelby here, underscores the fact that in the United States, public health is a state and local affair. So, there's not a national public health department. Like the CDC can make guidelines, but it does not have state or local public health authority. And so, there's a lot of different jurisdictions, and they've really been under a lot of stress during the pandemic, and they've been kind of attacked for doing the job that they're supposed to do. So I think that's something that we can all do, is advocate for strengthening our state and local public health departments. I mean, we've been very fortunate to work with the state lab and with PHMDC, and they're outstanding. But it's very difficult to and we're lucky, I think, to have a very supportive climate in Dane County, but that's not true everywhere. And then for just a couple of examples to talk, to put some meat on the bones of what we've been talking about, obviously viruses do not know about geographic boundaries. And so, throughout the pandemic, we have been involved, all of us in different combinations, in, outbreak investigations that span multiple jurisdictions. So somebody went to a concert or a meeting in Minnesota, and then they traveled through Wisconsin and visited some friends here, and then they went back home to Illinois. And, they were just spreading SARS-CoV-2 COVID along the way. But that means that instead of being able to kind of track that outbreak through just, the set of people who interacted, those people moved across multiple state and local public health jurisdictions. And so just being able to share information to understand, is this a variant that we need to be concerned about? Suppose this is an emerging new variant that, all 3 state public health agencies would like to control. Just being able to share that data across those agencies is very difficult, for reasons that are kind of based in totally understandable and I think important considerations about privacy and independence of different jurisdictions. But it also really hampers the ability to rapidly respond to a quickly changing situation. And similarly, I think because there isn't like this sort of national push, I mean, we're seeing some sort of modernization strategies that are led by CDC now, as Kelsey alluded to. But when state and local public health agencies want to gather richer data that they can analyze themselves, they're often forced to, as Brittany said, either farm it out to a company that doesn't do public health or build something themselves with very limited resources. And so instead of having, dynamic data systems, you might rely on somebody just like manually entering data into a table. And so right now, I'm the last person on this call who should be talking about this from like a data science perspective. But when we've been tracking the infections with different variants across Wisconsin. And we'd really love to have that information together, like in a place where we could search it from the public health side at the state level. But as Kelsey was saying at the beginning, the way we decide like what is Omicron and what is Delta, that itself changes. So, she mentioned there's this machine learning algorithm that sort of like goes through all of the global sequences and kind of parses out like, okay, this looks like a new lineage and we should give it a name. And like, oh, wait, actually, these viruses over here, they actually should also belong to this lineage. But the ones that I said last week belong to this lineage, now I've decided they're different and they should go over here. So what we call Delta and Omicron today is not going to fully capture all of the possible viruses that might get named Omicron in the future. And we might even decide 2 weeks from now that a handful of viruses that are categorized as Omicron today really should be better called something else. And so if we have kind of these static approaches where there's somebody behind the scenes manually entering data into a table and it's not dynamic, then we've had a lot of internal discussions about like, is it useful then at all to save this information? Or are we actually doing ourselves a disservice when we have to like go back to that fossil record that Kelsey was talking about and parse out, well, like back in 2021, like when did Omicron take over and how fast was that? Well, it depends on what you call Omicron. So the integrity of this data is also something that like really needs to be considered more carefully as we go forward. And the more we sort of silo data into smaller and smaller jurisdictions, the harder it is to maintain that integrity.
Oh, so maybe we're entering a little bit into an age of fatalism with the pandemic, with the fatigue and the exhaustion that's here. And I'm sure many of those who are attending today have felt this too. But it's very easy to sit on a panel discussion of any sort or a think tank and the answer is, well, we should have more resources. The reality, though, is that in an exhausted public that's sick of having spent $16 trillion already on the pandemic, the appetite for providing those resources in the future might be pretty limited. So I guess I have a two-part question for anyone on the panel who wants to answer it is, one, what do you think things are going to look like a year from now? And two, how do you make changes and improvements if there aren't more resources forthcoming?
I will say a few points to this. I think one being every decision that we're making in the laboratory is this sustainable? Is this something that we can carry through as funding declines? Historically, we've kind of seen these trends where when a pandemic occurs, we see an increase in funding, and then afterwards, as people kind of forget about it, or things return to some sort of normalcy, that funding then declines. And so everything we're doing is trying to look at how do we approach our situation today where we're trying to get to where we want to be, but also how do we build this in a way that's sustainable? And I think, again, kind of going back to these ideas of some of the new throughput technologies, some of the new sequencing approaches. When I walked into the laboratory 4 years ago, I said, sequencing is going to completely take over. Like, why are we even still doing PCR? And everybody kind of laughed at me. And now we're talking about how do we get funding to build a whole new sequencing wing, because that's where the future is. That's where we're going to be going. And it's, I think, a question right now is how do we get the most value out of every dollar that we spend, and how does that value, how do we grow that sustainably? And those are definitely questions that we are asking every day.
Does anyone else want to take a stab at that?
Well, I was just going to add, I guess, a couple things. First of all, is that with respect to the money that we've already spent or that we've allocated, my point before was simply sort of readjusting perhaps how we distribute those funds to do more things like building the infrastructure instead of just running tests. And so I think that could be evaluated. I don't know all the details behind the way that the money is distributed, but I think ensuring that we're doing, that we're building not just for dealing with the now, but also dealing for the future is, I think, important. And then had a totally separate thought about what Kelsey just said about the sequencing wing. And I'm going to have to sit on that thought because I've totally forgotten it. So I'll pass it on to someone else.
Go ahead, Tom.
Well, I think what I would add here on hopefully a more hopeful note, and this is not just trying to weasel out of the what do you do with fixed or declining amounts of dollars question, but you know, one of the things that has really been like an unexpected and pleasant surprise for me is the way that that at least aspects of Congress have responded to this pandemic. And so I think, you know, as you guys well know, most of you on this call, you know, we've been really lucky to have very strong support, particularly from Senator Baldwin, but like more broadly from Congress, that I think finally realizes that, you know, the kind of boom and bust cycle responding to previous infectious disease outbreaks and pandemics that Kelsey was describing is like not is not smart, right? So, you know, just in the past 10 or 15 years, you know, I guess if we go back 20 years all the way to the original SARS, you know, there was a flurry of activity, you know, around research around emerging coronaviruses and you know, all with kind of this sort of under the rubric of pandemic preparedness. We should worry about coronaviruses in the future. And then people working on coronaviruses started to lose funding as you know, there wasn't a sustainable source of funding for that. And we saw the same thing around you know, avian influenza viruses with the 2009 influenza pandemic, and then Ebola, and then Zika, and so forth. And each time there have been a growing chorus of voices saying that we can't just sort of respond to the current pandemic, the current outbreak, you know, where there's like a slow kind of lumbering response of increased resources, you know, which tends to meet the current outbreak too late to really affect it. And then only to have that dry up and not be ready to respond to the next pandemic. So I think there's been a lot of discussion that's like serious for once about, instead of just responding to COVID and providing resources to track SARS-CoV-2 variants and to surge testing, which is important. But there's also, I think, a conscious effort, which is gaining bipartisan support now, to provide, you know, at least 10 years worth of sustainable funding for the kind of like pandemic preparedness, the detection, you know, in individual cases and in the environment, at the animal-human interface that we've been talking about today, as well as a piece of training the next generation of scientists who are going to need to sort of bridge these gaps between the laboratory and public health and data analytics. And so I hope that actually gets translated into real, tangible, you know, deployable pandemic preparedness measures, as well as training for people, for the kind of human component that Shelby was talking about. What's really going to happen, I'm not sure, but I think at least we're seeing movement in a direction that I haven't seen with seriousness before.
Shelby, you said you'd remembered your thought?
Yeah, it's that public opinion is very strong, and in public memory is very short. So thinking about how public memory is very short. Maybe some of you guys saw Michaela Schifrin fall yesterday on the slalom, and you're like, oh, this is terrible. And then there's all this news about what has happened to her. But you know what? Everyone's going to forget about it in like 3 days, because public memory is really short. And so people 5 years from now, maybe they will have forgotten about the extreme situation we're in right now. Maybe they won't. I'm not sure. But public memory, I think, is very short. But their opinions are very strong. And so I think that trying to, you know, convince people that public health is important needs to be done. It's like we need a PR campaign for the public health agencies because right now there's just so many conflicting opinions. People who want masks, people who don't want masks, and they've got their strong opinions why one of these matters over the other. I think that we need to have, like I said, I think a PR campaign for the public health agencies to remind people why these exist, why we need to invest in them so that that can be persistent.
Well, so I'm going to wrap us up a little bit early, I think, unless this last question goes longer than I expect. And I'm going to actually build on that and ask, one final question, which is, in each of your realms, imagine the year is 2027 and we have a new SARS-CoV-2 coming out of China. Let's just say Vietnam, so is not to say China, different place, mix it up a little bit. We have some of the same basic dynamics as we had in early 2020. From each of your perspectives, what do you do differently next time around so that the impact is minimized? So what have we learned and what would we do differently? And we have a bunch of different roles here. So first I'll turn to Kelsey to present sort of the state lab view. What would you do differently if confronted with a similar type of threat in the future?
So that's a good question. I think from a public health standpoint, there's a few things that we're working on to kind of support whatever the next pandemic might be. One of the things that we're really keen and interested in is a pathogen agnostic sequencing approach. So can we sequence viruses without having to know anything about that virus? Currently, all of our sequencing approaches require us to have some sort of primer or idea of what that genomic structure already looks like before we can start generating sequence data from it. And so having this agnostic approach means that we could start sequencing samples faster, looking for this virus well before there's any like kits available or anything like that that's commercially done. On top of that, I think, having a fast track to testing. The state lab was one of the first labs to begin coronavirus testing in the state. And, there's definitely a huge initial upfront demand. And I think we learned a lot of things about how to handle that situation. And I think going forward, there'll definitely be a lot more to kind of stand up that capacity, stand up that throughput, sample management, sample handling. How do you get results out of the lab that we learned? And I think that our response will just be that much better the next time this rolls around. I think from a public health standpoint or a laboratory standpoint, those are the kind of two main areas that we're most focused on for the next pandemic.
Brittany, from local public health perspective, what do you think?
So we actually, last summer when cases got so low and our mask mandate went away and some of us were getting back to our pre-COVID jobs, we did some after action reporting where we kind of reflected on how the pandemic response went and things we would do differently. And so we were having a lot of these conversations within our data team. So we have a lot of different things. A lot of them would be more at systems levels or just, wishing that the CDC would be better funded and having a more forceful response at the beginning. But I think for us, we would like to more rapidly follow and learn from other countries. I think that because public health is so underfunded and we have very little nationwide systems here in the US, it's been hard to learn a lot from the US. There's places like the UK that have, national healthcare system where they have really strong surveillance of cases and sequencing, and it's national, and so they can make better statements about what they're seeing. And there was just a lot of information coming out from countries at the beginning that I think we could have done better at learning from and tailoring our response based on that right away, rather than waiting on guidance from the CDC or guidance from DHS and being more slow to respond. So that is one thing that I think we would do differently. Another thing specific to our data team that we've talked about is, and this can kind of go both ways, but some of us would have liked to be a little bit more accessible to the public during the response. I think there's been a fair amount of loss of trust in public health or in various governmental entities. And we would have loved to be more responsive and be able to answer questions and be more transparent and explain how we were thinking about things and why we were making certain decisions. And sometimes that wasn't allowed. Like we were interested in doing like a Reddit AMA, like Ask Me Anything for the data team, but that kind of got shot down for us. But we were interested in trying to just give good information and respond to people who had legitimate questions and concerns. And I think that it's so polarized now, it's really hard. And there's a lot of questions and accusations that happen in bad faith. But yeah, I think if we had really built that trust and just better relationships towards the beginning, that could have served us better in the long run.
All right, and I'll divvy up Tom and Shelby. Tom, if you want to talk about academic labs and sort of individual level sequencing, and then Shelby, you can talk about environmental sampling. That might be a good way to break this up. So what would you have done differently, Tom?
Well, I think from an individual lab standpoint, I think that we try to get involved early and work with partners like you and to develop a response that used our strengths as researchers but could also benefit public health. I think it's difficult to say 5 years from now what would be kind of laboratory level things that I would do differently. I think sequencing technology, data analysis technology and approaches are all going to evolve. I think kind of as Brittany was saying, what it boils down to really is kind of personal and institutional relationships that build trust and the ability to share data. And I think the biggest challenge that we had in trying to stand up a genomic surveillance that was both responsive to the needs of genetic epidemiology and just trying to figure out how the virus was spreading, in individuals in Wisconsin, and report that back to our public health colleagues, but also use that opportunity to see, like, can we take this data and make insights about how the virus is evolving that will also help us forecast what will happen in the future. These are activities that are highly regulated, that, as you well know, Dave, like there was no clear jurisdiction about who should make regulatory decisions about what data gets shared, what data gets reported to whom, by whom. And even though people in academic labs who are developing new methods for sequencing pathogens, new methods for analyzing the geographic spread and molecular evolution of pathogens that could have direct impact on the public health response, and like need to understand from public health partners, like what is the kind of information that would be most useful to you. Like these two sides really want to work together. There was just a ton of red tape that got in the way of that. And so what I would do differently is keep the lines of communication that we established then open, and try to get our institutional brethren on in all of these different silos to understand the value that we have hopefully gained from working together, and combining our forces, so that the next time this happens, we don't spend 3 months fighting over, like, can I say that a person was infected with Omicron in Dane County versus Milwaukee County? I think we can share that information without betraying, individual level identifying information. But there's a value to the, public health that trumps sharing that, that makes it reasonable to share that information. So I think these dialogues need to continue. There needs to be institutional trust. How we build up that public trust that Brittany was talking about, super important. Don't know how to do it. So I'll stop there.
All right, and finally, Shelby.
So I guess I'll kind of add to what others have been saying. I've written some notes down to myself that how, I felt like people came together early, but there was no like action, as Tom had just said. We had to wait several months to get through all the regulatory tape to figure out what the heck we could actually do for anything. And so we needed more of a national coordination system, as Brittany pointed out. So with respect to those same challenges associated with environmental sampling, I think we have to use what we've learned and not to forget those things, and be nimble and flexible across all the environments where we think pathogens may lie. And I think importantly, as Tom said, that we need to engage with the public and have them trust us. That is critical. So one of the main challenges we've had when trying to collect air and look for viruses in the air is people say, well, my ventilation system is great. We don't have pathogens in our air system, or we don't want to know if there's virus transmitted in our school. I'm just going to stick my head in the sand and not know, because if I don't know, then it's not there, and that's great. And so we have to make sure that the public doesn't want to stick their heads in the sand. And if a new pandemic comes out in 2027, and in people doing environmental sampling say, I'd like to know if there's virus in this airport, or I'd like to know if there's virus in the waste, like in water systems, maybe even like the lakes, not necessarily wastewater, or, in various soil conditions, or whatever it is, that the public needs to be flexible to let investigators or public health agencies explore those different environments for pathogens, so we can more quickly figure out where to be looking to be on alert for these different pathogens. So the public needs to ideally not stick their head in the sand, and be flexible and nimble to work with those of us who are trying to figure out what's going on.
All right, thank you, Shelby. So with that, I'll turn it over to Brian for some closing remarks from the organizers, and thank you all again for attending.
Okay, thank you all. Before I give my closing remarks, I just want to say there are a lot of data scientists being trained at UW and throughout the UW system. There's 700 undergraduates in data science right now, and it keeps growing. And there's lots of interest from the data science community. You didn't get a lot of questions, but you have a number of people listening in very carefully today.